Hello student, we will start the cytoplasmic crossing over. In this topic, that is CO2 sensitivity in drosophila. CO2 sensitivity in drosophila. Inheritance of sigma particles in drosophila. The fruit fly drosophila are sensitive to CO2 and they can be immobilized by exposing them to CO2. The effects of CO2 concentrations on drosophila was discovered by Heritier and Tichier in some of the strains of the flies. These flies are much more sensitive to CO2 than the normal and are affected by much smaller concentration. It is found that if these flies are exposed to higher concentration of CO2, soon these become anesthetized and finally are killed. Reciprocal crosses between normal drosophila produce different results. Parent sensitive female, capital S, and normal male, small s. That is the phenotypes, capital SS and small ss. Gametes, capital S, cross with small s. F1 offsprings, capital S and small s. All CO2 sensitive cross drosophila offsprings. When CO2 sensitive females are crossed with normal males, all the offsprings are sensitive as shown in the above cross of them. The female again transmit sensitivity during successive generations when crossed with non-sensitive males. On the contrary, when males of the sensitive stock are crossed with normal female, only a few of the offsprings are sensitive. This shows that more sensitivity to transmitted through the female parents. This is the proof that the sensitive characters in drosophila transmitted outside the chromosomes and can possibly be associated with the cytoplasm. These self perpetuating units present in the cytoplasm and most probably associated with CO2 sensitivity have been called as sigma. The heat labile particles. Inheritance of Kappa particles in paramecia. This inheritance of Kappa particles was discovered by Sonneborn and his colleagues. They found that some cytoplasmic particles, that is Kappa particles, and their relation to nuclear genes in Paramecium aurelia, specific Paramecium aurelia with killer stain. Specific Paramecium aurelia with killer stain is able to destroy other red of paramecia by secreting some toxic substances, paramecin, into the water in which they live. This type of individuals is known as killer, and the paramecium in destroyed is known as sensitive. The paramecia of killer stain contains large number of particles in their cytoplasm known as Kappa particles with DNA components and are able to self replicate. These can mutate also and can divide, duplicate independent of karyokinesis. Killer paramecium with K gene in the nucleus. Sensitive paramecium without K genes. The Kappa particles are cytoplasmic particles and strictly transmitted through cytoplasm. Their production is controlled by a dominant gene K present in the nucleus of killer strain. Capital K present in the nucleus of killer strain. Sensitive strain possesses its recessive alleles small k. Nucleus with K gene, capital K gene, maintains and produces. Cytoplasm with Kappa particles gives rise to secretion of paramecium by killer type in water, destruction of sensitive paramecium. With the help of specific procedure, it is possible to cross the individuals of killer and sensitive races. The killer clone is carrying the K gene which is dominant, while the sensitive variety possesses small k. When killer capital K conjugates with sensitive small k, the exoconjugates are capital K, small k. These according to their genotype should be killer. In the ordinary case of conjugation, where only the nuclear material is exchanged and there is no exchange of cytoplasmic material, it has been found that each exoconjugant gives rise to the organisms of the type that is sensitive produces killer. This suggests that killer characters is not governed by genes. In the second case, when the conjugation is prolonged and cytoplasmic breach between the conjugation is larger than usual permitted exchange of cytoplasm, all the offsprings are killer and their cytoplasm contains visible Kappa particles. This means that the Kappa particles from the killer strain are transferred to the sensitive strain through the exchange of cytoplasm. All the offsprings are killer and their cytoplasm contains visible Kappa particles. This means that the Kappa particles from the killer strain are transferred to the sensitive strain through the exchange of cytoplasm, so that sensitive individuals has also been converted into the killer. Therefore, capital K, small k individual may be either a killer or sensitive depending upon where it has killer clones. It has received Kappa particles or not. It is possible to convert killer clones into sensitive by making them to undergo very rapid fission. Cytoplasmic inheritance. Cytoplasmic inheritance is seen in number of species in the organism. There are two separate genetics chromosomal system in nucleus, extra chromosomal or cytoplasmic genetic system, which is primitive one found at the early days of the emergence of life on earth. In the course of evolution, cytoplasmic genes coalesce into the more easily bundled chromosome of the nucleus. Both the systems are independent to each other. It can be confirmed from the replication of chloroplast and mitochondria. So cytoplasmic inheritance is significant as it provided greater flexibility for growth of certain organelles of the cell. This is the CO2 and Kappa particles in the cytoplasmic crossing overs or cytoplasmic genetics. Okay, thank you.